Hey, I'm Elian, and today I'm going to turn a pile of leaves into protein. When you think of protein, you probably think of food, meat like steaks or chicken, or plant-derived protein like lentils or chickpeas. But proteins aren't just something we eat. They're a critical component of all biological life. And that means we can find protein in stuff that isn't food, like fur or feathers, or, like we're going to see in this video, leaves. In fact, for the average plant, the leaves contain more protein than the grains or the fruit, which is why spinach and kale are considered high in protein. They're literally just edible leaves. But we can also extract protein from less edible leaves, like crop waste, or even the leaves you could find on trees. And the process to make this happen is actually really cool, and it has applications to food security, world wars, and even how we might survive a nuclear apocalypse. Now, if you've watched any of my videos before, you know I can't spend that long learning about something without trying to build it in my basement. So that's exactly what I did. And in this video, I'm going to take you through that process. The substance we're going to be extracting is known as leaf protein concentrate, or LPC. And our first step is to get some leaves. Now, for reasons I'll explain later, I was most curious about extracting LPC from crop waste. So I contacted a friend from Microgreens Ottawa who was kindly able to give me some microgreen waste for this experiment. My next step was to wash the leaves out, to remove any soil or other debris that were attached. Then I laid them out in a tray and dried them off for about 15 minutes in my food dehydrator. Then I weighed the leaves, which came out to just around 50 grams. And this is where the extraction really begins, because our next step is to rupture the leaves and obtain their protein in the form of juice. The majority of leaf protein is stored inside the cells, specifically in the form of rubisco, the main protein that is used for photosynthesis, and incidentally, the most abundant protein by mass in the world, because there's both so many plants, and all these plants have so much rubisco. Since the protein is trapped inside the leaf cells, we need to break open those cells to get the protein out. So to do that, I took my leaves and chucked them in a food processor. The blade slashed through the leaves and left this pile of shredded leaf matter. So now these crushed leaves have their proteins released from their cells. But in order to separate that protein out, we first have to get rid of the solid leaf bits and get just some leaf juice left behind. And at this point, you might be looking at what these shredded leaves look like and be thinking, well, this looks like making coffee or tea. And these shredded leaves do look a bit like the inside of a tea bag. And you wouldn't be wrong, because like when you steep tea, you're getting flavors from the leaves without drinking any of the actual leaves. But unlike when you make tea, the juice in this case, which contains the protein, is coming directly from the leaves themselves. But fortunately, we can still use the same tools that are used for these hot beverages. A coffee filter. Coffee filters are perfect for this because unlike most filters, they're designed to absorb as little water as possible, because that's the part that goes in your coffee. In our case, we want to maximize the amount of leaf juice we extract, so we want a filter that will stop all the solids but let as much liquid through as possible. So, by wrapping our shredded leaves in a coffee filter and squeezing it into a cup, kind of like wringing out a sponge, we get a nice, green and solid-free cup of leaf juice. And as you can see, the resulting solid matter is now mostly dry and a lot more crumbly. Okay, so we've gotten rid of all the solid stuff that's not protein. But the juice doesn't just contain protein. It also contains other soluble parts of the cell that were also released when we shredded it. Getting just the protein out of the leaf juice is the hardest part of this process, and it's also where most research in this area has gone. In our case, we're going to exploit the fact that protein precipitates quickly under heat, a lot faster than anything else in the leaf juice. Different papers report different data on how much and how long to heat this juice, but the consensus is that it's somewhere between 70 and 95 degrees Celsius for about 10 to 15 minutes. I figured I would stick on the high end of both, to make sure I precipitated as much protein as possible. So I plan to heat at 90 Celsius for 15 minutes. So I set up this small bowl as a boiling pot and added a thermometer so that I could try to keep the temperature around 90 C. But basically, as soon as I started the heat and the temperature reached 90 degrees Celsius, the protein began to precipitate and was over within like 90 seconds. My guess is that in other papers, researchers were using a much larger volume of liquid, so it took much longer to reach 90 Celsius all the way through. In any case, the precipitated protein rose to the top of the bowl and formed a green layer. But as you can see, when I move that layer aside, the liquid beneath was brown. This is actually a very good sign, Previous papers I had read had all described a brown liquid being left over after the protein was filtered out. So seeing that gave me confidence that the heating process had actually worked and gotten the protein to precipitate out of the solution. Now the next step was to filter the protein, and I set up another coffee filter to get rid of the brown liquid, and slowly but surely, the liquid filtered off until there was nothing but green protein on the filter. But filtration can only go so far. So once I'd filtered as much as I could, I put the entire coffee filter inside my dehydrator and dried it for 6 hours. And 6 hours later, I returned to find a coffee filter with completely dry, green leaf protein concentrate. When I weighed it, accounting for the weight of the coffee filter, I had obtained 0.16 grams of protein. So there we go. Leaf protein concentrate extracted from microgreen waste. Now admittedly, the extraction efficiency here is super low. From 50 grams of plant material, we got 0.16 grams of protein, which is an efficiency of 0.32%. Now granted, the plant is probably only around 2 to 6% protein, because it has very high water and fiber content. So we did recover most of what was available. But this can be optimized, and there's been actually a lot of research on how we can make this process more efficient. In fact, the idea of extracting leaf protein concentrate dates back to World War II, when food shortages in Britain prompted researchers to look for literally anything that could be turned into food, leaves included. In the decade following the war, significant investment was made into learning about this extraction process, which was led primarily by Dr. Norman Pyrie at the Rothamsted Experimental Station. But ultimately funding died out in the 1970s. There has, however, been a revitalization of this work with regard to nuclear war. In a nuclear war scenario, our biggest concern isn't necessarily the nuclear bombs themselves. Although they are devastating, it's the fallout afterwards. Specifically, a rather likely outcome is what's known as a nuclear winter, in which the ash and debris scattered by these bombs rises into the atmosphere and literally blocks out the sun. And without the sun, we basically can't produce any food. In this situation, we would need to extract everything we can from the plants that are left on Earth, which is where leaf protein would come in handy. It's a research focus of the non-profit group AllFed, which has done much more recent work on the LPC production process. But of course, we really don't want a nuclear winter to be the only reason to work on LPC. And fortunately, it has other applications, namely for turning crop waste into animal feed. Much of rural Africa is dominated by smallholder farms, which have some crops and some animals. Feeding these animals remains a challenge, particularly when grazing areas aren't readily available. After a harvest, crop waste, like the corn husks or the wheat stalks, are fed to animals, but these can only be used for the short time during the harvest. This means that around 80% of that crop waste is just left on the field, because the animals are fully fed. In other words, for a few short weeks a year, farmers have an excess of animal feed. If we could take this crop waste, process out the useful part, in other words the protein, and store it, we could convert that excess into a solution to the shortage. That's why I enjoy this experiment. It's a micro-experiment that shows us how we could solve some of the world's biggest problems. And with that, I hope you enjoyed my return to experimenting and making videos about it. So make sure to follow on LinkedIn, subscribe on YouTube, and as always, I'll see you in the next one.